Welcome everybody to this webinar. And I am Raghavendran. I'm a research consultant and bioinformatics mentor with Pine Biotech. And I will be your mentor throughout your research fellowship program. So I'm very excited to share with you how this program works, and we will answer some specific questions related to what is this program about and how to join the program, how to submit a project on OmicsLogic, and what are the general structure of the program that will help you perform your own bioinformatics research with us. So this program can be roughly divided into three main stages. So at the first stage is a training stage. So this is a stage which sets the direction for participants or students like you to define in which area or in which research field that you are going to work on. So some of you might have a very clear idea on which field or which research topic that you want to work on. And some of you might have also had some introduction to bioinformatics as a course or as a curriculum or as a different introduction means. So, but some of you also would like to gain more knowledge before making a good decision. So for those who are still deciding and who still are not from a bioinformatics background, we always suggest you to start with two very basic courses, that is Introduction to Bioinformatics and Bytes and Molecules. So these basic courses are designed to introduce the basic concepts of bioinformatics and its application to different research fields or areas that employs various omics data types, which can be subjected to multitude of analysis methods. So we are introducing you several methods. So by the end of these two introductory courses, you should be able to understand what are the different methods employed for different omics data types. And also, you also should have a clear idea of in which field or in which data type or in what question that you want to work on. Not question, at least a focus on which direction that you want to move. So at the end of the first stage, you should be able to, for example, have all the necessary skill sets to identify a specific research question that you would want to work on. So in this stage where you are going to identify a research topic, so you will have to first find a data set that has the potential to answer your research question. So more on this a little later. So in the next stage is where you actively pursue your research, discussing results, inferences, and all the necessary suggestions and feedbacks from mentors all along, right through. So after these three very important phases, you will have to prepare a presentation that you will submit in our portal, the OmicsLogic portal, as a project, so that you can showcase this project and share with your network of peers and share your poster if you have prepared in conferences and in meetings, scientific meetings that you can attend. So let's again go through for example, sorry, I think it's not stopping at one point. Yeah, so we throughout the course of this program, we have a weekly progress report form that you will have to fill up and submit to us, and you will have to discuss your progress with the mentor at least once in a week. So these progress reports are excellent way to evaluate how you progress through the research fellowship program. It is helpful for you and also helpful for us to find or to place you where you are currently, and then to help you in at all the necessary steps of your research progress. So our learning resources for this program includes various courses that cover different specialization fields. So we have specialization fields like that focuses on the bioinformatics parts of the omics field or omics data type that you would want to start with. Like if you want to work with metagenomics, then you can start with metagenomics courses. If you want to work with transcriptomics, you can start with transcriptomic course. That both of these courses and also other ones goes in depth in the type of data that you are going to analyze and the various different options of analysis methods that you will employ for different questions and different answers that you are pursuing from these data types. So you can also decide on different data science skill sets that you want to start developing so that you can apply to various omics data type in general. So for those who would want to go through skill set development fields or specialization, we have multiple coursework that explains or that introduces you to Python environment and R environment and different data science applications and machine learning, which includes machine learning and deep learning methods. So all of these methods can be generally applied to various data types that one can obtain from several analysis methods that are designed specifically for different omics data types. So one example of how these courses are designed, let's take a look at designing a bioinformatics research project courses. So these courses are designed to take you deeper into those specific respective topics in a step-by-step process that are outlined into separate lessons. So for example, this is a course which we suggest to all the participants so that they can understand the necessary steps involved in how to design a bioinformatics project. So more on this a little later. So also, throughout these courseworks, we have several practical assignments along the coursework and example projects that takes data from publicly available resources and the data that are used in popular research articles. And we will design separate analysis pipelines or separate analysis methods that is going to answer a research question that is not straight, that is not related to what was the focus of the research paper. So we will take the data and then try to combine different analysis methods in employing or in using these publicly available resources to answer the research question on our own. So while you are going through these example projects, you will get a good understanding of how to take a publicly available data and how to create your own analysis methods that is going to answer a specific question or a hypothesis that you will form from basically understanding some introduction about the data as described in the associated paper or publication. So these different exemplar projects are designed to include a variety of topics like in epigenetics, human health, agriculture, virology, immunology, precision medicine, etc., a lot of different ones. So also microbiome also. So these practical exercises and these projects will give you necessary exposure to understand the data-driven discovery of real research behind these projects. And it will also encourage you to think critically about how to go about designing your own projects and what are the necessary ingredients in creating a successful bioinformatics project. So as I mentioned, the basic purpose or the fundamental purpose of doing these practical assignments is to get a deep understanding of data specific challenges and also to showcase how various methods of analysis are applied in real world situations. So when you compare the results that you obtain with the one that are focus of the publication, then you can clearly understand how the questions are formulated to get to the publication level from such a publicly available data or such a vast quantity of data that is dealt here. So this can be your project at the end of this analysis also. So once you have acquired all the necessary skills and you are performing your research in a good healthy progress, and then you can use these skills to apply them to various different research problems that shapes the bioinformatics and biological research of today. So it can be from precision medicine, it can be from population health, can even be space and astrobiology. So we have various example projects that that are categorized under such different categories. So it will give you all the necessary introduction to take the data from these specific fields and then apply your own analysis methods for the question that you are interested in trying to ask from those data. So let's start going through how would you find a research topic. So let's take a practical approach to how to get or how to identify a research topic. So as we said, the major steps that are important to design a research problem that we have been discussing so far is first you have to frame the problem, and then to frame the problem you have to understand what is the latest current trend in that specific problem. So you can, by framing the problem, like you can try to narrow down that I want to work on cancer, or you want to be even more specific. I want to work on, let's say, head and neck cancer. And then you can understand the literature, or you can go through the literature, review the literature, the scientific results for the last 5 years or even last 10 years on this specific topic. So once you have gone through the literature, you can then identify that specific gap. So the literature review should tell you what are the areas that are addressed more frequently or more in a very detailed manner. And also it should also give you a hint on what are the areas that are not covered generally. So you can try to approach that area or try to take that specific area and then collect a raw data that will, or you can actually find publicly available resources, data that is necessary to answer your question or your problem, or answer the gap in literature that you have identified so far. So once you have collected these data, then you have to create a processes or create a sequence or create a pipeline for the analysis, detailed analysis of this data. And then you start by exploring the data to understand the patterns, and then you perform in-depth analysis that you have designed for the data. And once your analysis is complete, then you infer from the results and then perform necessary biological implications by relating it to the characteristics of the sample or to the nature of the problem that you started with. So for example, in head and neck cancer that I started with, I might have started with trying to categorize the tumor tissues. So if I can collect the necessary data that will help me in categorizing or classifying the tumor tissues into multiple categories, then I have to find whether my analysis substantiates this claim or substantiates this hypothesis. And once my analysis and my inferences are complete, then I can put together this as a story and communicate the results to our peers. So let's start how to find a topic or how to choose a topic. So you always start with topics that you know about or topics that you care about, and etc. For example, this could be a personal reason, or this could be a reason that you are deeply interested in. So you can start by noting down, or actually by collecting few slides or few information about the background, about the topic background. So then you have to start preparing or start spending few time in analyzing or researching the topic. So how much has been known? So what has been studied, and among those what is studied, what is left to be studied, and what will be interesting in this topic that will be interesting to the audience and that will be interesting to you also. So you have to do something that you also care about. And then once this is narrowed down, and then you have to find a data that is going to answer the question that you can choose from this in this topic. For example, if the topic is very specific or very narrowed down, you might not find sufficiently studied data or sufficient volume of data that will answer your query. Or if the topic is too wide or too broad, you might find a lot of data and you will be left with a lot of choices to make a good decision. So you have to find the right balance that that will maximize or that will help you in identifying the data, perfect data that will answer the query that you have in your mind. So how to do this? For example, so finding right questions or right sources are one of the issues. So this is always a back and forth process where, for example, in the original research, you take the raw data and then you perform the analysis and then you compile different analysis and then you find a solution. Or you can also go back, you can take from the publicly available resources and collected data, compiled databases, and then you explore for the possible trends. And then from these trends, you can actually design a question, whether the initial question that I have in mind can be substantiated by following some of the trends. So we will get an example for this in a while. So for example, if you to narrow down on the topic of research identification or topic identification, you first go through the reviews, and then you can categorize those reviews on their analysis methods or different questions that they are answering. And then once you have a thorough meta-analysis done or thorough analysis done on the reviews that are available or a publication that have done for the last, let's say, 2 or 5 years of the data, then for your topic, then you can identify the unanswered questions, and you can identify the gaps, and you can also identify some of the debates and controversies which are left unanswered in this topic. So these are the areas which you need to focus on. I mean, on the areas which are obviously answered by many people, you don't want to reinvent the wheel. So we want to identify those areas which are not covered a lot, and which has a lot of potential to improve the science, or which has a lot of potential to fill the gaps in the science in that particular research area. So that gap and how to answer, how to design specific analysis methods to answer those gaps and answer those unanswered questions will define your hypothesis or question.